Fine. So I'll send you a recording. Fine. No. Okay. Sorry for that. I thought it was when I didn't notice that. I just started sharing the screen. Apologies. Right. So I was saying that let's start to. If you look at the tensor, for example, in the ventricles here, you can see big spheres. Okay. Why? Because again, we are in the CSF, high diffusivity, isotropy is the same. But if I go from here to here, you have, again, a spherical shape, but it's smaller. Why? Because now we are in the thalamus. In the thalamus, there is stuff. There are neurons, there are tissue, there are membrane, etc. So the diffusivity is decreased. So the shape is the shape is the same, but the size is different. Now, if we go from here to here, it's not only the shape, the size has changed, also the shape. Now we have the cigar shape. So the axial anisotropy is came before. This is because why matter, we see fiber. Clearly, if we start talking about size and shape and visualization like that, people get confused. This is not a very synthetic way of describe diffusion information, right? So this is just used to do pretty picture in paper or do more kind of more mathematical description. So what we need is to do some matrix we can use for our study. And the first matrix is mean diffusivity, okay? Which is essentially the average of the three eigenvalues. So the first simple number you can do in science, you do a mean. Okay, first thing you do is always reporting a mean of something, your descriptive statistics. Okay, mean diffusivity, that's exactly the mean. And what it's doing is completely ignoring the shape of the tensor. It's just telling you how big is the tensor. So what is the size? How much is the average displacement of water molecule in a fixed amount of time? And you see, for example, the CSF is high diffusivity, and the rest of the brain is kind of much lower. What is interesting here is that a B1000, both white matter and gray matter, has kind of the same diffusivity. And it might seem, and that's why I think this matrix was less popular, has been less popular than FA, we'll see in a second, because it's boring. There is no contrast, well, there is no anatomy, what I'm using for. But to be honest, this is not a bad news, because if you have a flat image, you will immediately see something that should not be there. You immediately see changes in diffusivity, for example, it's changing cellularity, or it's changes in edema, for example, amount of lipids, so you can see edema. For example, this metric is very sensitive, even more than FA, to detect neuroplasticity. Yeah.
I think I'm a bit confused.
Sure.
Maybe it's just stupid.
No, go ahead.
If I look correctly, so EWI is basically.
Oh, that's a bit okay.
Yeah, sorry.
Okay.
Just realized I didn't understand. So DWI is our acquisition, and then there is ABC.
Yeah.
I mean, I don't really understand the relationship with mean diffusivity.
Okay. What is
the
the difference between mean diffusivity and DC? I'm coming to that. Okay. First of all, we collect as a raw data from the scanner DWI, diffusion weighted images. Okay, clear. And we have a B0, the T2, essentially is by the way, it's called B0 because it's also a DWI. It's weighted in diffusion with weighting equal to 0. So essentially there is no diffusion there. Okay, there's no. So that's why I say T2 or B0 is exactly the same thing. So when I have these two data, I can do the simplest analysis is to just kind of normalize and do a little bit of maths and extract an ADC map. Okay?
So it's a manual reconstruct, it's not a concept style.
No, the ADC is exactly, the ADC is the map of diffusion. So the raw data is the WI and the B0 is another raw data, and the ADC is the map of diffusivity. That is the simplest. So it's a monodirectional or the average of 3 axes. When you do the tensor, you do something different. When you do the tensor, you have at least 6 DWI or 30 DWI. So you need to measure diffusion on 30 direction in space, because the diffusion is, as I said, is a 3-dimensional. With this, say, 30 direction and let's say 3 or 4 B0, you do again some maths. And what you get, instead of one number like the ADC, you get the tensor, this 3-dimensional form, okay? Now, from the 3-dimensional form, it's difficult to work in kind of in tensor space. What you do, you generate a new map. And the map is the mean diffusivity, which conceptually, You're very close. Conceptually, mean diffusivity ADC represent the same thing, but ADC is only in one direction. Mean diffusivity is the true mean diffusivity across all directions. Okay?
And so we also said that ADC is basically the average, no?
No. Yeah, that's you have to complain with clinician, not with me for that. Because in radiology, people call ADC the average of 3 ADC. Because they do ADC on X, Y, Z, because they want to remove the artifact of anisotropy, and they just do the average ADC. So I know it's a bit confusing, but that's how we should call it mean ADC, okay, to be technically precise. But what they do a DWI and they do an ADC. Why they call it like that? Because the scanner automatically do the mean for them, so they don't know. Okay. Exactly. It's just the average of 3 axes. So this is pure clinical scan. So this is not, you're never going to see this in research. So on the scanner, when you do a clinical ADC or clinical DWI, you just get an average automatically of three axes of diffusion, so that you remove any anisotropy and you can see an approximation of mean ADC. To be fair, mean diffusivity from the tensor and the mean ADC should be theoretically the same. It's not for other reason, other model, etc. But in principle, they represent the same thing, the average mobility in the voxel. So the mean ADC on 3 axes or the mean ADC on the tensor should be fairly close. Sorry, let me check the term. Okay, we're fine. Okay. Yes. No, I didn't say magnetic.
Value. I mean, in my mind, there is the magnetic field of MRI that and then there are some.
There is magic. There is magic. Because if I'm going to explain you the physics of diffusion, that would be half an hour on its own. Okay, so what I'm saying is the B value is the contrast that there is a way in the sequence that I can make the image sensitive to the mobility of water molecule. How much sensitive is this B value? The higher the B value, the more the contrast. Let's leave it here for now, because if we're going to talk about neuroscience, emergent property, you don't want to go in MR physics, because of why we die here. Okay? Okay. Right. So let's go back to what we're saying. So now we have the tensor. We have the 3 eigenvalues. We calculate the mean. As I said, this is a kind of flat image, but it's very sensitive, for example, for neuroplasticity or for, for example, change in cellularity, oedema, stuff like that. That is actually probably the most sensitive map of all the different maps you can get for this diffusion. It's just an average. As I said here, the beauty of that, because it's a full 3-dimensional model, it takes in consideration the orientation, because we just have the 3 axes. So when we calculate the mean, what we get is a rotational invariant index, like I was saying a few seconds ago, the mean diffusivity from the clinical scan. Okay, but but I can do more with the tensor. It's not just the mean. Otherwise, there was no point to do the tensor. That's what we can do. As we do the mean, we can also do the standard deviation. Okay, so we can actually see how the 3 eigenvalues are different between each other. Which said in a much simpler term, I can see how the diffusivity is different in different direction. Okay, so don't worry about too much to the equation, but essentially the top is a variance, and the denominator is a normalization done in a way that FA is always going from 0 to 1. Okay, so this is not a big difference. Mean diffusivity is an absolute number of telling you how much is the diffusivity of the tissue. FA, people not always realize that, is just a ratio, sort of, because it's relative. I can have a very high diffusivity one axis and axis, or can have a much lower diffusivity axial and all, and I can always have the same FA. As long as the ratio is the same, I always get the same FA, okay? It's a relative number, the FA. And why is useful? Because it tells us how organized is the white matter. For example, we have very high anisotropy when fibers are parallel, like for example, the corpus callosum, we have getting closer to 1. When we go more lateral, we can see lower anisotropy because the fibers are less coherent, start to fan out, there is crossing, etc. FA is lower. So FA is a measure of the shape of the tensor, okay? And like mean diffusivity, also this index is a rotational invariant. So it doesn't change if I put my scan in sorry, if I put my head in the scanner and I change the position. So in theory, I should always get the same number, because if I measure it along enough direction, I can actually get always the same number, okay? As I said, to get a good rotational invariance, you need a 30 direction, because otherwise, if you have less, you might have some kind of statistical bias due to noise, blah, blah, blah. But 30 direction up, the number is very reproducible. And that's how you use your tensor. It's important to understand when we talk about diffusion, we are not looking at a molecular staining of myelin or a molecular staining of neuron, okay? So if you're familiar with histology, you have staining that's specific to protein, specific to specific antibodies, etc. Diffusion is not specific. That is one of the big complaints many people do to diffusion is aspecific. Diffusion is just a metric or something, it's a tool to measure the interaction between water molecule bouncing on structure, on tissue. So what we are looking is how water moves in an environment, and from the way the water is moving in the environment, we cannot guess some information about the environment itself. It's a probe, it's a mechanical probe, okay? So, for example, if I want to see if a tissue is very well organized or disorganized, I can see, for example, that a very parallel fiber and compact fiber might have a higher FA, and a disorganized bunch of fibers, for example, low FA, okay? And maybe, as we see before in the mean diffusive map, mean diffusive can be the same, because we see that between gray matter and white matter, mean diffusive is not much different at B1000, okay? But also, you can also look at changes with diffusivity. Imagine you're looking at babies, and you look at the brain development. You see the increase of myelination over time. So you have a system, for example, axon, with not much myelin at the beginning, and when the brain do to maturation, you have more and thicker axon. So in this situation, your axial diffusivity is not going to change. So along the direction of fiber, the obstacle is always the same. But perpendicular to the fiber, now you have more obstacle, you have more myelin sheet. So essentially, this tensor is becoming slimmer compared to here. So what's going to happen? Essentially, FA goes up when you have more myelin, and mean diffusivity goes down. And that's exactly what happened in brain maturation. Less mean diffusivity, more anisotropy. And that's what's been reported by many. But now we can do the opposite. Let's change from neurodevelopment to neurodegeneration. Let's imagine we have MS. We have the opposite. We lose myelin. What's happening here is we go from here back to a situation where we have less myelin. So we read exactly the same way. FA goes down because we have less obstacle radially, and mean diffusivity goes up because, again, more space for water molecule to move radially. Surprise, surprise, check literature, and that's what's happening. Okay, so when you use diffusion, any model, you always have to think about what the what the water molecules are doing. Okay, so that is the key for understanding how to use diffusion. Is always the water molecules. Not there is no staining, there is no, because sometimes people say, oh, FA is a marker of myelinization. FA is the marker of, oh, mean diffusivity marker of neuroinflammation. It's sensitive to, but is not specific. Okay. Now we have other maps, and for example, we have this map here. For example, lambda 1, 2, and 3. As I said, they are coming from the tensor. You have the three Euclidean value, and you can just get normal map of that. Whereas, if you remember, I told you lambda 1 was always the biggest, and you have see bright internal capsule is very bright. Corpus callosum is very bright because it's telling you what is the diffusivity along the fiber. Okay, and in fact, some people told lambda 1 is called also axial diffusivity because it's telling you the diffusivity along the fiber. Then we have another matrix called radial diffusivity. Okay, and radial diffusivity is the average of lambda 2 and lambda 3. And what is telling you is essentially how much is the radial component. Now, this is really important to understand that this is working not everywhere in the brain. This is working where our assumption of coherent fiber orientation. Imagine you have a bunch of fiber crossing the same voxel, okay? And we'll see later how this is kind of a big problem when we do tractography. You have crossing fiber, and then you calculate the tensor. Where is the axial diffusivity here, or radial diffusivity? The tensor is the average of what's going on. So the axial diffusivity probably is the vertical direction, okay? Let's say it's the 30 degrees crossing, okay? And the radial diffusivity is here. So axial radial to what? Okay, so that's why axial diffusivity have to be used with careful. So when you have a very strong dominant orientation, yes, you say on average, this is the axial diffusivity of this component. But when you have a lot of crossing fiber, careful to use. That's why mean diffusive FAs are more common in the study. Now, let's have a look specifically why I was saying we have limitations. So the tensor, when we look at the diffusion in a, what can I say, in a coherent bunch of white matter fiber, we can see this, what we call diffusion propagator, the displacement water molecule, is parallel to the fiber. Sorry, is identical to what we see with the tensor. So the tensor is a good model for this diffusion propagator. But when we start to look at crossing fiber, you see the propagator is not described at all by the tensor. You see, we always have an ellipsoid, even if this is the real displacement water molecule. Okay, this is the reality. This is the real diffusion. This is how we model with diffusion. So we have an ellipsoid, and the direction of maximum diffusivity is not the real one. So now you can understand why tractography, go, for example, from the wrong direction, create a new track, or stop, okay? The same thing here. That's the example I showed you before. So this was a very quick introduction to DTI, okay? Now, what's happened after how can we go beyond DTI? Well, for many here, we were using DTI for doing both tractography and microstructure. And in one way, people try to push the best to get the maximum out of it. So if I want, for example, like me, I was working on tractography mostly. I wanted to get the best possible tractography, and I want to use sole crossing fiber, etc. So people started to, with the limited amount of time you have on the scanner, for example, 10 years ago, or even a little bit more, people only can acquire 60 direction for research, forgetting going to 100 to do Multishell. It was impossible to do because there was no multiband, okay? Today is different, but at the time, 60 direction was kind of the realistic limit you can do for clinical research application. And for example, people do 60 direction, B2000, 3000, and we're doing spherical deconvolution, Q-ball, etc. On the other side, people started to think about, I want to look more at a more complex way water move inside different tissue. For example, I want to separate different compartments. And we have what we call microstructural imaging, okay? And these require something else. They require different behavior. But again, we are limited. We only have time for 60 volume in total. So people decide to sacrifice angular resolution to resolve crossing to get more shell, be a shell, 20 direction be 1000 and 40 direction be, 20 direction be 700 and 40 direction be 2000. I'll come back in a second, I'll explain why we need more to shell in a second. But that is a kind of, it was a bit split in this community. So from DTI had two brothers. You have the microstructure and they have the tractography. And they were doing different things. So different acquisition, different project. And for a time, if you want to, you have to choose either one of the two, or you can add two different data sets. Today, things have changed. Today, we go from, for example, 20 minutes for 60 direction. Now, with the introduction multiband, we can do 4 minutes, we can do 60 direction. So with multiband today, we can do, for example, B500, 1500, 3000 with 30, 60, and 90 direction, so 180 direction, 180 volume in about 15 minutes or less. And this is something you can do now research. Okay, before we're doing 50 minutes for 60 direction. So now we are starting to converge back. So now that we are developing method that you can do tractography and microstructure at the same time. So for the last few minutes of this lecture, we're going to talk about Multishell and advanced microstructure modeling, okay? Again, to explain what is multi-shell this is a very simple example of what is a single shell acquisition first on the left. Single shell is to measure diffusion along different direction, but with the same b-value. So you can imagine conceptually, if the b-value is a radius, you have the same diffusion contrast along different direction. That's it. And that's what we do for DTI, spherical deconvolution, and many classical methods. But then, if I want to get a better characterization of microstructure, I want to use different weighting at different contours. So I start to look at different direction, but also different b-values. So you have different, you can imagine different shells, okay? 500, 1500, 3000, for example. And that's what that's what microstructure modeling is using today for doing their stuff. So what we mean for microstructure imaging. Now, when we talk about microstructure imaging, we start to look at more in detail at the real biological tissue. As I said before, the main limitation of diffusion imaging, diffusion tensor imaging, ADC also, is that it's not specific. It's giving us information that what is the mean diffusivity, what is the FA, but as I said, FA is not myelin. FA or mean diffusivity is not inflammation. These are metrics that correlate, are modulated, but they're not specific. So one of the attempts of microstructure imaging is trying to identify, develop models that can be specific or some specific biological characteristic. I'm not saying they're doing great. No, I'm not saying they are perfect. So they're doing great. Sorry, they're doing great, but they're not perfect. But is a good improvement compared to a more general description. Okay. So why we need multishell? When I say multishell, so people completely blank and we disconnect from my lecture. So now I explain why we need multishell. When you use the tensor and we use the classical model diffusion of the ADC, we assume essentially that diffusion is a simple Gaussian process. So you have a water, certain amount of time. You can imagine the displacement of water molecule follow a Gaussian probability, simple. Which means if you look at the signal different b-value, it's just following a simple exponential decay, simple. So if I want to characterize this exponential decay, I just need one number on this line. By knowing the b-value, by knowing the line, I immediately get the diffusivity. I don't have there is no other combination because it's a single exponent and I can get the D by a single measure. The problem is, if you look at multi-compartmental model, you have intraxonal water. Water inside axon is often modeled like fully restricted. Okay, so there is no possibility for water molecule to go through the myelin. When I say restricted, you can imagine you can approximate like impermeable diffusion. So even if you have more and more time, the water will stay there. So it doesn't go beyond a certain limit. That is the meaning of restricted, which is, again, clearly this is not a Gaussian, because with a Gaussian distribution over time, the displacement will increase. And then you have extracellular diffusion. And then you have, I don't know, macroglial diffusion. Then you have astrocyte diffusion. Then you have so many stuff around. So different components can contribute in different ways. So if, for example, we just assume two compartments, you see that attenuation is clearly deviating from a normal exponential. So now a single measure, a single b-value, cannot tell you if you have just this, which model are we. So that's why you need at least 2 or 3 points to try to guess which one the attenuation. So if it's 1 exponential, 2 exponential, 3 exponential, blah, blah, blah. So that's why you need multiple b-values, because you want to decompose the different attenuation profile of the different compartment. That's it. Okay? So if we do that, the first thing we can do is what we call diffusion kurtosis. And diffusion kurtosis is essentially the next step after the tensor. Because if you have, with the tensor, we assume everything Gaussian. With diffusion kurtosis, we're just looking how much my displacement, the mobility of water molecule, deviate from Gaussian. Kurtosis, is the higher order of a distribution. So if it's a Gaussian, my kurtosis term is equal to 0. So Gaussian perfect, the tensor model is perfect, there is no problem. But if kurtosis is greater than 1, my distribution is more pointy. What it means if it's more pointy? It means there are more restricted compartments in my, so there are so much more restricted compartment that water is trapped. So more water is around the center, so it's not moving far. There's only a little tail. Compare a classical Gaussian system. So by looking at the kurtosis coefficient, it's like looking at a measure of how much restriction do I have in my tissue. Okay. And it's quite interesting metric. It's becoming it seems to be very, very sensitive, for example, to neuroinflammation. And let's give you some images. So here the classical tensor, and now you have different kind of, I think for me at the moment, the most interesting is the mean kurtosis. Sorry. Is the mean kurtosis because it's telling exactly inside the voxel what is the average amount of restriction. No, I'm okay. Sometimes people call about fractional anisotropy kurtosis. I don't like it. It's usually people think the mean kurtosis is the same as mean diffusivity. No, because mean kurtosis is telling about restriction. Mean kurtosis is more similar to an FA term of anisotropy restriction. Okay, so that's why you should use mean kurtosis for analysis. Yep.
Sorry, I'm not sure I get how you come from like one single measurement to a distribution, like in this case. Is it only from the Multishell or?
You have, you measure two, as I said, if you have a Gaussian distribution as a model, you just need one acquisition, one sample, okay, because Gaussian distribution means exponential decay. Non-Gaussian distribution means multi-exponential decay.
I see.
Okay, so that's the link. Okay. Now, there are other models more advanced. For example, some model like DSI, Q-space imaging, try to sample many b-value, many b-value, many direction, and you can do a transformation of the signal. And for each box, you get the actual distribution. But for that, you need to have a lot of approximation at the end. This is very blurry. You will see later when we talk about crossing Fibre. You don't resolve very well. Or you have method like MapMRI, that actually give you something that is similar to the propagator, but you get metrics that are coefficient of non-Gaussianity, return to probability, is some measure that is difficult to link with biology. Okay, it's a nice descriptor of the shape of the propagator, of how the water moves, but linking to biology, pathology is quite difficult. I haven't found many applications, clinical or neuroscience, that they actually use it. What has been much more successful are methods like NODDI, white matter integrity, etc., where effectively what they do, they literally define a signal for each compartment and try to fit a different compartment. Now, I'm not gonna present all these different models. I'm just gonna use this slide here as an example, because we can actually put it together to a single model. So initially, CHARM, it was the first microstructural multi-compartmental model. What I was doing was literally modeling the intraxonal water as restricted and extraxonal water as hindrance, like classical Gaussian diffusion. The sum of the two diffusion gives the composite hindrance and restricted diffusion, CHARM, okay? And the idea is you have a tensor for the hindrance, and you have a very sharp, very restricted signal for the intraxonal space. Over time, they also say, well, let's put another model for free water, and that is the ISO component. Now, we can replace this with a distribution of, instead of single compartment, we can have distribution of sticks, like imagine many axons, which with this diffusion only one axis, and this with this fanning, and you know what you have? It's called NODDI. It's exactly, you want, for example, don't look at this at all, only restrict, so hindrance and free water, and what you get is free water elimination at the eye. So what we realized that by playing around with plugging in and out a different compartment, we have what we call today the standard model of white matter. So there are few papers search the term standard model of white matter. It's a bit heavy mathematically, but what it's telling you, essentially, you can divide different compartments with different equation, essentially, and then you feed the different equation different way. Then you have NODDI, free water, white matter integrity. There are different variations on the same thing, okay? So you can do many things with the same data. So as long as you have multi-shell data, you can fit different model, and each one of them have different characteristics. What is important to realize is this. Realize that the sensitivity is different for each matrix. When you go higher, probably this is the only thing I want to look at is this graph here. This one, actually, this one is the best. The more complex is your model, the less sensitive, or the more sensitive to noise. It's obvious. If you have 30 measurements and you fit one parameter, you get one stability. If you have 30 measurements and you fit 15 parameters, clearly you will be more sensitive to noise instability, okay? So by using the most advanced model, it's not necessarily always the best thing to do. You need to be balanced between the quality of your data and what is the research question. So for example, I'm still a big fan of the TI and diffusivity FA because they are extremely sensitive. If I have a strong hypothesis, I don't need to separate the different tissue and the sensitivity is there, I'll go with the TI. But if I need a little bit more to understand exactly where this change is coming from, yes, I need to go for a higher model, okay? Not necessarily bigger is better or more complex is better. So think about that. Now, I want to conclude very quickly with a sort of announcement that this is something happening basically now. In London, we are installing now this new scanner. Probably you're familiar with the connector scanner in MGH. It's been installed now, literally in October. Sorry. Hopefully it will be available. It will be ready by the time I go back to London. It's a head-only high-performance scanner. It's a 20 millitest like the connector in Cardiff, for example, other place, but it has 750 slew rate. If you're not familiar what it means very fast. In the sense that it's so fast the API distortion, susceptibility distortion are almost not visible. So that allows you to go much higher resolution or even compress a lot of your TV. For a while, it will be the only in UK and Europe. So if you're interested in play around with this in this scanner, let us know. We actually want to offer this as a European Brexit permitting system. So collaboration, we don't ask to be author or something like that. If you just want to be, if you want to write a grant and want to be more happy to collaborate with you or not, and then you can use it. And just to annoy people at St. Thomas's, nobody's here, I guess, we can do better than 7 Tesla and 9.5 Tesla, because for some experiment with diffusion, you need very high p-value and very short echo time. And you, at the end, on the back of the error calculation, show that we have more SNR than a 9.4 Tesla. So in terms of SNR, we can really push a lot. And then, of course, you can do something like something that we do in preclinical. Here was, I think, 2 weeks ago. This is the big minus going inside the old CNSD. If you remember, Steffi, this is the old CNSD coming in. And now this is the picture I got from Friday. This is I don't know if I can see the right number. Oh yeah, here's a 2.16 Tesla. So we are ramping up. We are ramping up the gradient now. These are all the power going into the scanner. We are charging up the magnetic field, so we're almost there. Hopefully, we'll not quench before I'm back. And just to show you what we hope to get, this is clinical 2 millimeter. And this is where we're hoping to go as a research stuff, 760 micron in half an hour in vivo. Yep. Yes. Let's call that. Yes. And last year, SMRM, they presented 500 micron isotropic in vivo under less than half an hour. Yeah, sorry. I finished, by the way.
Thank you. Sorry. Can it be used in clinical?
We have a grant for it. Well, we submitted 3 grants. 2 got rejected, 1 went through. So we need to resubmit now because they're interested now, finally. And the idea is to apply neurosurgery. Yes, because I think now that we could offer something new to neurosurgery, because it's like putting a better pair of glasses now, because if you try to understand where is the tumor here, you cannot not see the clear boundary between track. Now you can clearly see where it's located, which if for example, if the tumor is between two track or not, or or if you even can see infiltration in better detail. So that is the goal we're hoping to get. As I said, let me know if you want to collaborate. We're open for collaboration and grant writing. And yeah, that's my job here is to get this kind of running. That's that's as a part of the new lab name, Neuroimaging and Biophysics Lab. I cannot be responsible for this kind of, so it's my baby, so he better do well with it. Okay. Okay, so question. We said before that with the tensor, we have different sizes, certain shape, we have mean diffusivity, we have FA, but we also have directions. Okay, now if we look at this zoom of the spring, we see that this cigar shape are actually showing a trajectory. So if you're now going to use the principal eigenvector, the direction of the maximum diffusivity, and you can actually see that with this information, we can actually guess where the fiber are going, essentially. But before doing tractography, there is one thing we can do very quickly. We can just put a color on our FA. So if we decompose, for example, sorry, noise. If we start to be hot, just let me know, open again. If we decompose the principal eigenvector around three axes, X, Y, and Z, and we get a color of each axis, for example, blue for Z or superior inferior direction, red left and right, and green anterior posterior, this kind of vector can be projected so we can have the different component. And if we give that, our FA can get a color like that. Now, you see an image like that many times, but what it means that with red, for example, we clearly have fiber that are going fully left, right. It's fully on the axis, okay? When you have the blue, we have fiber that are going superior, inferior along the Z-axis, green, anterior, posterior. And of course, we have different other degree of colors depending how the different angle. Why this representation is important? This is important because already neurosurgeon, even before doing tractography itself, they're already using this map because they actually see already how the anatomy is being displaced. And you can actually see something that with FA, you can see, for example, if you look here, oops, sorry, too much. If you look in this region here, it's not clear to understand which track or bundle is passing by, but if you look at color, you already know clearly that there are at least 2 or 3 different trajectories, red, green, and blue at the same time. So probably there are different pathways going the same region. So they already help you to think. Well, of course, it's not as easy to read as a tractography, but it's already, you start to have a directional information, you can start to think with it. But what we are here, of course, is because we want to do tractography. And the simplest way to understand tractography is to, again, take our principal eigenvector here. Can you see my mouse? Yes. So if we select if we select one point in our tensor field of the brain, and what we do is simply generate an algorithm that integrate step by step a trajectory that somehow, we'll see in a second which algorithm we can use, but we can interpolate this trajectory based on the different tensor field. We want to reconstruct the underlying kind of white matter orientation. So remember, with tractography, we don't see axon, we don't see fiber. We just see eigenvector, or just that big box, 2 millimeter, 3 millimeter, that tell you in this box, the trajectory, this one. That's it. Using only this information, we try to predict what's going on under, what is the underlying real white matter orientation. Just following this instruction, we can start to generate a lot of streamline, do some dissection, and this is what we're getting. This is just basic DTI with probably, simple rules. For example, I'm just using a FA mask to say I don't want to go outside the brain. That is a good rule for tractography. And I use an angle. Say I don't want to turn more, let's say 45 degrees. At each step of the algorithm, I continue to propagate only if my next step is within 45 degrees, because I want smooth trajectory. I don't want to change sharp trajectory because I'm looking at big tracks or smooth kind of changes of direction. With these rules, you can dissect track like this one. This is the whole arc with fascicular system. You can go Broca, Wernicke, etc. And it matches quite nicely with the Klinger dissection. But Klinger here, you can see this particularly they are kind of amplified with the scalpel. Here you see this string tube. They are just amplified with the amplified using some kind of lighting effect, shadow, because string tube, look at this kind of rendering, are good to give a nice three-dimensional representation. If it was just a line without shadow, without lighting, it would be just a blob of red. You don't see any three-dimensional image on that. But what is also important to realize that the individual streamline here is not a biological entity. It's not a real axon. It's not a real fiber. Remember, it's a mathematical construction of the whole field of tensors. So the individual streamline starting here, stop going there, might be not real. What is probably real is the overall shape. We have something starting from here, going here, some kind of a structure of U shape on the back. That makes sense. Think about, always, to my student, I always tell that, think about the weather forecast. When you look at, for example, yeah, we are in France, we're near also, well, no, we are not near the Alps here, but, but imagine, okay, you're close to the Pyrenees here? No, not even. Anyway, so I'm from Italy, I'm from Milan. Alps are near, are from where I'm coming from. And imagine when you see the weather forecast, you see the wind, the line of the wind coming down the mountain. Okay, you see the line of the wind. But this wind is not the real wind that is going to happen for you, right? So this is just a mathematical presentation. You know the wind is starting from the mountain, going down to Milano, then going to Venice. Okay, this is kind of, that is not the path, the real, there will be some weird path, there is some kind of changes over time. The same thing is tractography. Tractography is a macroscopic representation of what's going on. So you have a lot of axon in the same voxel, a lot of changes. So we know an average is going that direction, but maybe the average might be distorting the real anatomy. So the real mathematical reconstruction, the correct mathematical reconstruction is not the correct anatomy, okay? This is important to realize. So you need to know your anatomy, you need to validate the things. Over time, we started to be confident with some track, but some track cannot be trusted just blindly, okay? Now, I don't want you to remember these different methods, but this is just to give you an idea that even with DTI, the simplest algorithm, there is not a single DTI tractography algorithm. There are many. The first one, the fact from Susumu Mori, it was a very robotic one. It was basically going straight inside the voxel, changing the direction when you change the voxel, and very robotically it was moving around. The problem with that is the step size was different every time. So sometimes you can go a very long step, sometimes just a little bit. And also you can just have a big branching just because you just miss the border on one voxel and you go into another. So it was very kind of, I would say, spiky, very noisy looking, the tractography, but it was one of the first. Then we start to introduce what we call now interpolated streamline. Essentially is a different integrators approach, like you have a Euler, Runge-Kutta, different mathematical way of essentially at each step, I interpolate all the neighbor voxel to calculate a smooth approximation that change over time. I don't have always the same direction. So I take into account the presence of other direction. And this equation is the very simple one, but you can get more term, etc. You can play around. So different algorithms, they'll give you different results. You can bias more to be more kind of bendy. As soon as you find a new direction, you can try to turn even more, or even, where is it, the tend. The tend was the first approach to try to solve crossing fiber in a very original way. I like it. Basically, you say, when I don't know where to go, down my head, go straight, and then I pass a voxel, look around, is there still anything that makes sense to my direction? If yes, I continue. So basically, there was a kind of, he has a sort of inertia. If you reach a voxel with low anisotropy or uncertainty, he would just going and hope for the better and continue. He was solving some crossing. He was a way to solve crossing, but that was the first one of the first approach. Okay, but let's take to the standard general interpreted streamline. If we just do that with a bit of dissection, already in 2002, Marco Gaetani was able to dissect this track, and are very close to how they were being described by the German neuroanatomists in the 18th, 19th century. And the big difference was that while here you need to do a postmortem dissection, up there you can actually ask a PhD student to do on a scanner. So it's much easier to do this. And in terms of acquisition, we started with, again, initially 30 direction was taking maybe 20, 30 minutes. Today, as I said, it might take probably 2 minutes scan with a multiband sequence, something like that. And this is as I said before, this is a reasonable acquisition if you want to do just basic DTI. You don't have time for anything else, just sticking this. Although we can have a chart discussing about tractography unions versus fMRI unions. I'm from the diffusion union, so I found really unfair that for fMRI you dedicate 20, 30 minutes of experiment for diffusion, no more than 5. So I'm always proposing for 10, 15 minutes also for diffusion. So that's if you want to have a debate, I'm here. But anyway, so if this is the case, so 5 minutes is what's usually is kind of a typical limit for tractography or for diffusion, but usually if you can do 15, 20, I can tell you the benefits are there. And then why tractography is so special? Because tractography is starting to show something new. It's starting to show, for example, that there is something across different subjects. When we do neuroimaging, we normally normalize voxel to MNI, this kind of stuff, but you see there is a lot of variability. The same track, this is an experiment we did many years ago across different sites, and we have some traveling heads going around scanning on different sites. And this is, for example, the sites of London. And you can see there is a big difference of 3 segments, the long segment of the acrophysical for 3 subjects. Now, if you look at 2 other sites, you have Cambridge and Oxford. London, Cambridge is exactly the same type of scanner. Oxford, completely different manufacturer, different sequence, etc. Again, the anatomy is pretty well preserved if you look at the different subject. Of course, even the artifacts are very well replicated. So I'm not saying this is a perfect anatomy. What I'm saying is tractography is capturing something that is unique to the anatomy of the individual subject. So we are talking about something subject-specific, precision medicine, this kind of stuff, okay? So tractography is able to do this. And again, this is important because, for example, I think I'm showing a slide now that probably many of you are already familiar. So most of the neuroimaging is living in MNI space. We normalize structural, we normalize fMRI activation because we thinking, and I'm not saying it's wrong, okay? I'm not making any criticism. I'm just saying we just do that because it's easy to work with that. But, and this is, I'm sorry that Michel is not here because I stole one of his slides. Yeah, the average is not always the right thing we want to look at. This is exactly, so a few ladybugs, and Michel average, this you have to create the mean, kind of the MNI template of the ladybug. And you can see here, this is a very old slide from 2008, 2009, I don't remember. And you can see this anatomy is not there in any of the ladybug. Okay, so what's happening here? What's happening is that when we do an average brain, we are getting, of course, the reproducible part, but we are losing something. We are losing the variability, okay? And I think maybe Steffi would talk a lot about, okay, so I'll just stop here, not going further. But I just wanted to say, that is one of the benefits of tractography. Tractography is born to live in native space. Although we can do something in MNI to normalize, to have template atlas is fine, but we have to be careful not to lose anatomical variability from our data, okay? And so in tractography, because of that, we can have big track, small track from this, or big volume, small volume, just because the anatomy varies across subject. And when you do that, then you can quantify what is the mean FA on the track, or what is the mean Malin fraction. So, and the moment we have any maps, MRI you want, in the space of the tractography data set, you can sample it, and you can get a mean FA along the track. So you can use the track as a volume of interest to quantify specific, customized to the anatomy of the individual subject. Because imagine you do just the region of interest analysis in your normalized space. Your normalized region might not overlap exactly to the anatomy of each subject. So you really need, the benefit is to have exactly the anatomy of each track, of each subject with your own dissection, one by one. And if you can do that, then you can still do, for example, analysis. Like, for example, look at intersubject variability. Someone is doing lateralization before, so this is something that you can want to do. So you cannot use the same mask. You want to adjust. For example, here I have to look at different lateralization of the acrofasciculus, and this was just looking at his anatomy, and then Steffi applied this to see if the lateralization can be used as a predictor of recovery from aphasia after stroke, for example. That could be something can be done. But of course, the main application of tractography in terms of clinical application is neurosurgical planning, because now we have a tool, as I said before, on a single subject, we can actually see how the anatomy is different. I don't have to do an average. I don't have to immediately see the presence of a lesion in the insula displays my corticospinal tract, my tractography is deformed, so the neurosurgeon immediately see how should I apply. I don't know, you say you're a neurosurgeon, right? So you might not be interested in exactly the cortical termination, probably practically, but you're more interested to know how the segmentation of the tract. But if we want to do some kind of, someone else want to do a study of cortical connectivity, maybe it's a different approach. So tractography, I've kind of, there is different way of using tractography, okay? So you can segment track, for example, in this case, or you can look at how the different connections are. So this was a very quick, a general example of how tractography, just using basic DTI is used. But there are problems. And this is my way of presenting problem. This is essentially a way to think about tractography. So it's like a train. You go, follow your track, follow your eigenvectors in the case of DTI. But sometimes your eigenvectors that take you in the wrong place. Either you stop in the middle of white matter, or you turn when you should not turn. Why? Because as I said, remember, there are some limitations. For example, let me see here. Here in this case, if the data set is very noisy, I see the general shape. I also see a lot of spurious streamline around. And or you can see track that doesn't exist. Cortical, pons cortical, or even, I don't know, an S shape or something like that. These are track that well, don't look real if you know the anatomy, but they look like track, but clearly anatomically are not. We know they don't exist. Why? Because, as I said before, mathematically they are correct. They are following the rule we decide, but unfortunately, because the tensor is just an average, the average of the directionality gives us this kind of artifact. So explaining the reason why again. Well, the first reason is because if you remember, we're using very simple rule. We put a white matter mask, say stay in the white matter. If you go out, stop. That's the first. And also you have, as I said, 45 degrees, for example, 30 degrees, and nothing else. So the tensor is, everything happening within this rule is allowed, okay? But also because we have important issue. As I said at the beginning, DWI is an attenuation from a B0, from a T2. So by going down, you are sensitive to noise. The higher the B value, the more the noise effect. Spatial resolution. Your data set is not yet 700 micron, is 2 millimeter, okay? So whatever happened 2 millimeter is average out. So you can see one fiber, we'll see in a second, we see crossing fiber multi-direction, but you have to be careful that the average might not be always the right orientation, okay? And of course, we have model limitation. With the tensor, we cannot solve crossing, kissing, fanning, etc. So we will see that step by step. Let's start with the noise. The problem with the noise is quite straightforward. If you look at the picture here, you can see good SNR FA, bad SNR FA. Easy because you see this kind of dots. You clearly see the noise of this picture. But sometimes people don't realize that the effect of a noise is also on the tensor field. This is a simulated data, don't worry about it. You can see if there is noise, my principal eigenvector direction will also be affected. So it will be more shaky, more uncertain. So my trajectory will go more or less in the right direction, but will kind of deviate. There will be some bias. And because tractography is an integration, step by step, we build up error the more we go, okay? So even when there is a good signal, you see the kind of still moving it a bit. We are not attached to a single axon. We actually following the overall trajectory. And if we do this, we follow this trajectory, and here we can jump on another track, stop. So that's why artifact happens, okay? Sorry, why we went back? I don't know. Yeah. Now, how we can compensate for that? One way of compensate for that is, for example, to map this uncertainty. And there are different ways you can do. The first, probably the most accurate, is to acquire, instead of one data set, I acquire 5 data sets of the same subject. And then I can do kind of bootstrapping of the data. So I can generate, if I have 5 subjects with each one 60 direction, I can permute this 60 direction different times. So I generate, I don't know, 100 different data sets and see how my estimate direction is robust or not, how it change over time. If I do that, I can see that the deep web matter usually is much more robust that get closer to the cortex. Why? Because it phase lower, so a little bit of noise make you change the tensor a lot more. When it's a very sharp and pointing, you really need push to change the main or direction, okay? But there are different ways to model. So I don't want you to think about, oh, I need to know this. Not really, just you need to understand that we can model the noise, we can model uncertainty, okay? And if we have an idea how much is this error, this uncertainty, we can get to what we call probabilistic tractography, okay? So somehow, I don't want to specify which model, we have an uncertainty measure in each voxel. So we don't have a single orientation, we also can have a sort of a cone or a distribution of possible fibres we can take. So if again we do the same experiment with tractography, instead of having always the same streamline, we have only one vector, now we have a choice. So every time we run tractography multiple times, we are gonna pick a different possible direction. So if the uncertainty is small, more or less we get the same trajectory. If the uncertainty is big, we're going to have a lot of variability, a lot of spreading. And here on the left, you can see the probabilistic reconstruction thing with, for example, bed post X. This is exactly from the, I think, the training material they released. You can see sitting from here, pretty much we are reaching already the whole brain. So if I lose the tractography completely unconstrained, with a seed, I'm going literally everywhere in the brain. What I see, you can see the most reproducible trajectory, here's one, he's going to try it in a capsule, that somehow replicate what they see also with deterministic. The only difference is, yeah, probabilistic feel a little bit clear, is meant to fill gaps, okay? But at the same time, it's giving you a lot of more false positives, I'm saying, clear. For example, this is the important point, sometimes people get a little bit confused. The probabilistic map here is not the anatomical probability. It's not say high intensity means more true anatomically. Doesn't mean that. It just means it's a highly reproducible. This trajectory with my data set, with my algorithm, for example, look at this trajectory here, stop in the middle of corona radiata. If you believe me, if I rotate to 3D, you will see